So I'm going to be talking to you today about ERGA in the context of the Biodiversity Genomics Europe project, and about the workflows and standards that we have been putting together to deliver reference genomes for the BGE project. So for those who don't know, the BGE project is a joint endeavor between ERGA as the European node of the Earth by Genome project and IBOL Europe, which is the European node of the International Barcode of Life. We applied together to the same call of the European Commission of the Horizon Europe program, and got this project where we try to join forces to enhance the use of genomics for biodiversity across Europe. So we have a very large number of partners already in the project, but our communities are much larger. Altogether, they involve more than 200 and actually probably more than 300 or 400 institutions. But in the project, we have about 34 participating institutions. And apart from the European ones, we also have some external participants, such as the IBOL headquarters in Canada, the EBP headquarters in the US, and the VGP representation as well. And here in the map, you see in the little circles where our partners in Europe are located. And in red, these are the partners that have that represent the sequencing centers. So we have sequencing centers that are spread in 6 different countries and working together to deliver the genomes. So in the BGE project, we have 3 major objectives. One in capacity, so to establish the functioning networks at the European level. Second, in production, where we want to establish and implement large-scale biodiversity genomic data generation pipelines for the entire European community. And that's where we derive the standards and automated workflows that we are going to present to you today. And also we have the application aspect, where we aim to show how the application of genomic tools can enhance the understanding of biodiversity and support the actions towards or against biodiversity declines. So the project is very large and complex, and we subdivided, so trying to connect these both networks, we subdivided the project in this following way. We have 2 streams, one representing the barcode community and the other representing the genome community. And we have the pillars here that represent the workflow of reference data production. And that's where we overlap, like the 2 communities have like very similar workflows, even though of course the details are different in the field to lab, sequencing at scale, and data to application. So all the way to submitting the data and doing the first analysis of the data. And we have a last pillar of biodiversity applications, which then has case studies and other aspects, connection to citizen sciences, citizen scientists, and so on and so forth. And we have the work packages of support, coordination, communication, policy, and ethics. So within the ERGA stream, so the genome stream, we have been trying to standardize or to look for the points in the workflow that where standardization is needed and helps actually the democratizing of the generation of reference genomes. And we figured over time, with this complexity of different institutions across different countries with different infrastructures, levels of expertise and so on, we needed a balance between standardization and flexibility so that we could work all together in synergy, and then working towards the scaling up of genome production for the future. So then following here, the process of the workflow for reference genome production ending and the publication as the last step, we have established some points of, that are standardized or automatized, and some points where they're either made at this point manually, or they are flexible depending on who is executing them. So starting with the sampling and metadata acquisition, we have been gathering the sampling SOPs, guidelines, when they're not available from the sequencing centers or from the other projects. We establish certain guidelines, best practices that are then standardized, that are usually made available via Zenodo or some very open repository. But the sampling itself, it's flexible. We have the records, the metadata records through a manifest of metadata that is standardized and then broke via COPO, so that we can make it sure that all the metadata that has been established as a standard and necessary for reference genomes is being computed in a proper way. And that's when we start already the process of getting this into the public repository, so directly into ENA, where we form or we create the first biosample. So then we enter into the laboratory part, where we have protocols that are from different sequencing centers. Again, they are shared ideally through protocols.io. That's more of a work in progress. So where we are trying to make all these protocols available, many of them standardized, but with the chance of one nice thing of protocols.io is that you can add some exceptions or changes, little changes that you have made in the protocol. Even when you use a standard protocol, you can add some comments there and have them computed. So once then this data is produced, either for DNA or RNA, and the QC is done, the raw data goes again into the repository as a almost first thing, and in many cases, even before the assembly gets started. So after that, we get into the bioinformatic workflows, where we start to produce the assembly part. And these workflows, we try, the ones that we have been using or the community has been using, we try as much as possible to gather under the same repository in the workflow hub from ERGA, with workflows that have been implemented with Nextflow, Galaxy, or SnakeMake. And Tom is going to talk to you a little bit more about this platform specifically. Also, another thing that we have established as a type of standard, once these assemblies are performed and the pipelines then can be flexible, even though they have been made available, each sequencing center or assembly team chooses which way they're going to assemble the genome, but they have to generate a report that is standardized and that is going to help us in the reviewing of this genome assembly. And Tom is going to get into more details of this as well. So after we have done this quality control, that's when, again, we upload the assembly, so the final assembly to ENA, and finally the annotation can be performed if the RNA-seq is already available. The last step of this pipeline is the publication, where we are working towards, also in close connection with the Sanger Institute, having an automated genome report engine. And this is somewhere here. No, yeah. So the idea here is that some parts of this publication production is going to be automated by gathering automatically information from text that was inputted by the sample provider, or information that is present in Gold or data portal or in our tracking tool. So then we have these other aspects here that this is what Tyler is going to talk to you about. So all this business, so 7 sequencing centers in 6 countries, tons of sample providers coming from everywhere. We wanted to get rid of our Excel sheets. So Tyler Alioto then came with this great idea of producing an semi-automated tracking console that hopefully one day is going to be completely automated, but where the different sequencing centers and different teams can actually enter data about the progress of the genome assembly and annotation process. we have established also standardized that ERGA within the ENA or within the INSDC repositories is a child of EBP. And under then the ERGA, we have the assembly projects with the primary assembly, secondary assembly, co-biont if that exists, and the raw data connected to this assembly project from its species. We also have a public data portal which only when everything is finalized and in the public repositories, then you can see what is happening within, and this is not only for BGE, this includes the pilot and also all the different projects across Europe that are connected to ERGA with ERGA as the umbrella. And this was developed by EBI. Already talked about this. Yes, so this here is to also thank the people working with me. They are usually here, Tom Brown and Diego De Panis. Many of you don't know them, and now you know Luisa Marines, which is in the media, so our communications officer. And that's it. Thanks for your attention. I think I hand it on to you, Tom, now.
Thank you very much. Yes, so I will expand on two things that Camila brought up, which is really geared towards how we report and ensure that our assemblies are produced in a way that's reproducible and that anyone who wants to use the assembly knows that it's performed in a robust way and can quickly find out the quality of the assembly and if they want reproduce themselves. So I really want to highlight that this is a lot of work within BGE from Diego and the University of Freiburg and Björn Gröding's team, who's helped us in particular with Galaxy. So I have two questions that I'm going to address in this talk, which is if we have genomes coming from multiple partners with different technologies, there are a very diverse set of organisms that we're working on, and then each center has its own compute infrastructures or expertise. How do we ensure that these genomes are of a higher quality and are of the reference quality that the EBP is trying to generate? And then how do we report these outputs in a robust and reproducible fashion? So to answer this first question, we have many pipelines because each center has different expertise. They've developed these pipelines over a long period of time. They don't necessarily want to start using someone else's pipeline when they know they have one that already works on their system and produces good assemblies. So we say how you produce the genome is not entirely important to us, but the output should be of a very good quality. And we're aiming for this 6C Q40 or Q50 as we're moving towards, and I think is well established within the EBP. So with this in mind, Diego De Panis, who's the coordinator of the Sequencing and Assembly Committee in ERGA, went to the community and said, which outputs do you absolutely need to see in order to tell whether an assembly is of a high quality? I think lots of these tools will not be too controversial and are well known among this group. And Diego took all this information and said, okay, how can we report this in a way that someone can then take an assembly and then quickly see whether it is of the quality that we want? So he has been working with the University of Freiburg team to develop a standardized reporting output. In this case, it's a PDF where someone can run QC tools on their assembly, give all these outputs into this Galaxy tool, and generate, as he calls it, an ERGA assembly report, or EAR. And now anyone, if they want to know the quality of an assembly, can look at this PDF, read through it in a couple of minutes and know this assembly is very good, or this assembly is not so great and needs some work. So within the ERGA BGE project, we've been using this to cross-check the assemblies that are submitted as part of the project in a very collaborative and open process. So what we've done here is sort of reinvent peer review, but we've taken away the handling editor and replaced this with a GitHub bot. And we do this all via GitHub. So at the top, I'll start you up here. The sequencing team, once they've finished an assembly, they will generate this EAR. And the way they open the review process is they create a pull request to our ERGA GitHub. And then the bot is kicked into action and pulls from a pool of assembly experts, a reviewer and a supervisor, who are going to look at the assembly, the ear, and establish whether it's good enough to be assembled to ENA. At this point, the reviewer will then enter a dialogue with the submitter to say, there are some things that should be changed here, you need to remove some contaminants. And then the role of the supervisor is to oversee this, moderate the conversation, take over if the reviewer goes on holiday. And then ultimately, once both parties are happy with the assembly, merge this into the main branch. And then the submitter knows this is my point where I can say my genome can be uploaded to ENA. And I want to particularly highlight the work of Arash from Björn Gruning's group, who's developed this bot for us and has put us to shame in our lack of computer science abilities. The reviewers themselves are pulled based on a scoring system. So we have at least 5 reviewers from each sequencing center, and they're given a score based on whether, if they do a review, their score goes down. If they submit an assembly for review, their score goes up. And if your score goes to the top of the leaderboard, you're pulled in as a reviewer. And this way we try and keep it even so that people who submit a lot of assemblies also have to do a lot of reviewing. And if you do a lot of reviewing, eventually you should move to the bottom and not be called so much. And again, this is all automated through this bot that Arash has written with Diego's assistance. And what we're really proud of is that this is an entirely open process. So I'll show you here the entire review process of this lovely little mollusk, Arcanoa. So if your Latin is good, you know that we're even assembling biblical species. And here you can see that Joe Collins from the Sanger has come back with saying, here are some areas I think you should have a look at. And then what's really nice about this process is then the submitter can say, yeah, I agree with most of those, but there are some areas where I'm not so sure. And we can paste this conversation goes back and forth. And what's nice is it's all tracked. So this pull request, anyone now can go back and look at and see how this genome assembly came to be what it is. And then at the end, once the reviewer is happy, they approve the changes, and here the supervisor, Diego, merges into main. And this is the point where the assembly is uploaded to GenBank and is now the reference. And this now means that alongside a genome, you can also go and find the report. This is held in the GitHub, and also the full review conversation. This is in the pull request. And our aim is to have this permanently maintained and ultimately link this to every assembly that's produced as part of the BGE process. So alongside the data portal that Camila highlighted, where people can track the public progress of the project in terms of assemblies, one can also find a full QC report of every assembly that's produced as part of the project. And we think this helps keep the process open and gives context for every assembly in terms of its quality and usefulness. So we've produced a genome, and now what do we do with it? And how do we show to people how we created it? So we're doing this via the genome reports, and I'm sure you've heard a lot about these from the Sanger. And in order to document how each step of the genome is produced, we use this workflow hub space. And I think I've mentioned this before on these calls. This is a repository of workflows developed by the University of Manchester, who are a project partner in the BGE project. And we encourage, or we're starting to force every assembly team to publish their assembly workflow in this space. And what's really nice about this space, so here I'm showing the CNAG pipeline, not only are the full scripts available and all the environments and all the tools, but also here there's a citation. And this lists every author who is involved in the development of this workflow, as well as giving a DOI. And it means that when it comes to writing the methods section, we ask the centers to write a reduced methods detailing the software that we expect. But then if someone wants to, they can go into the workflow hub place and see the full workflow, all the parameters, all of the versions. And the people that worked on this pipeline are credited, even if they're not authors in the paper. And we do this not only for the assembly, but for all the figures that we generate as part of the, sorry, all your faces are on my screen, as part of the report. So over here on the right, we have a workflow in Galaxy developed by Diego. And here up at the top right-hand corner, there's a run on Galaxy option. So if someone wants to, they can click on this button, Run on Galaxy. It opens this workflow in their Galaxy instance. They can pull the data from this report and generate these figures themselves if they want, or perhaps more likely, generate these figures for their own genome. And this is certainly helping us a lot in not having to redo a pipeline every time we want to generate a figure and reduce our method sections, as well as keeping everything fully documented and reproducible. So I hope I've highlighted that we're trying to keep things collaborative, and this review process it's not only been good for QCing assemblies, but also it's been a learning process to help people in the curation process, which is a pretty complex task, especially with some of these funky little organisms that we're working with. But we want this to be open and documented for every genome. The workflows are useful not only for reporting and publishing, but also we think other people will want to use these in the future, and we encourage this. But this gives a full provenance of who performed what, using which tool, which version, and which pipeline. And we're pushing this to help us create genome assemblies in a distributed manner and fully reported. So if any of this sounds interesting, I encourage you to come along to a sequencing and assembly meeting. They're fully open. You can hear more about what we get up to from this video from Diego, but we ask you to register to be GDPR compliant. So with that, I'll hand over to Tyler. Thank you.
One moment. So there we almost are. Do you see the presentation? Yeah,
we do.
Yes. Good. Okay. Thanks, Tom. Thanks, Camila. I have a short presentation, hopefully, on some of the kind of internal development we're doing. And so in order to track who does what, when, and where, the reports are great, but we have to get there first, okay? So we're tasked with tracking the data going into assemblies. So I have a data production task, right? Need to make sure that we produce assemblies. We're under a tight deadline now with a milestone at the end of next month. And we're trying to get as many assemblies done as possible. And the idea is to figure out where we're at. And you can't do that from the public data portal because that only shows what data has been submitted. We need to know the status of all the data before it's submitted as well, right? So Camila alluded to one of the problems was a bunch of shared Excel spreadsheets or Google Sheets being passed around. You know, sometimes you have access to it, sometimes you don't. The formats in these things get rather funky, as you can play with colors and fonts and all that. And we had multiple spreadsheets tracking very similar data. And it is a real chore to keep those in sync across the different groups. So we have many countries, many institutions, many people. We have a work package structure thrown on top of this, complicating things as well. It's supposed to make it work nicely. But we have separate meetings for the different work packages. And these are for the ERGA stream. I'm only going to be talking about ERGA, not eyeball. The sampling work package, basically the sequencing, and then all the data analysis, right? Including assembly, annotation, and responsibility for how the data is moved around. We have distributed sampling, so we have hotspots in different countries, Spain, Slovenia, Greece. We have community sampling, where we're getting samples from external PIs, right? Providing the samples. That has to be coordinated. We have now bio blitzes. We have some case studies, all different ways of collecting samples, and we need to keep track of this nicely. We're not doing this all at one sequencing center, so we have to know where the samples are to be shipped, right, and how. So we have Sanger, SciLife, CNAG, Genoscope, the Italians, and Norwegians, and the assembly, right? So to simplify things, we'll have the assembly done mostly at the centers where the sequencing is produced. But sometimes we split the sequencing. So let's say Hi-Fi is done in Florence, but the Hi-C is done at Sanger, right? And then we have to coordinate who does the assembly at that point. Annotation, right now it's fairly simplified that Ensemble will do everything, but we also have a community annotation task led by Genoscope. And this has yet to be really incorporated into the tracking, right? Yeah, and on top of it, we have the public data portal, we have COPO, we have GOAT, we have all these other informatics systems, and we have to make them all talk to each other. Okay. So we needed a tool to allow this communication between the different sequencing centers. We all have our own laboratory information management systems, our own way of doing things. But we need to kind of share this kind of sequencing progress and assembly progress in some way. So we need to keep track of the species, prioritize them. Originally, we were getting the GOAT status because we had these spreadsheets being sent to GOAT, and then we get the status from GOAT. Now we're sending the information to GOAT. But we needed a management tool to assign who does what. This was all done kind of behind the scenes, and we're trying to make the tool be the place where this assignment can be made. So on and so forth. We need to know where the samples are, what the progress of the sequencing is, and facilitate submission of the data and really drive towards submitting this to the INSTC. And then it'll show up in the public data portals. There's a bunch of features that are still being implemented. We have some email notifications already, so that when sequencing is done, the assembly team gets a notification that the sequencing is ready. So enough about that. And how did we implement this? Well, I implemented it in Django because I've been using it before. It's what our LIMS is written in. It's a nice framework that provides a nice web front end and object models that basically correspond to database tables. So we can rapidly develop. I used it on another project, and I basically took that. It was working very well and applied it to this case. The idea is, well, I'm not going to go over the Django model right now. If anyone has questions, you can ask later. But what it allows us to do, it has a nice API REST framework, right? So we can, you can query the database programmatically from your own site, right? So this is ideal for sequencing centers to be able to automatically submit run data and sequencing status, setting up a script to do this so that you don't have to enter everything manually into the tracking system. We can pull information from the ENA. So currently I can track reads that have been submitted. Now assemblies, we're adding that so that as assemblies get submitted, they show up in the system as well. This kind of duplicates a little bit the data portal, but we need to know when stuff hasn't been submitted so that we can pester the people that have to do it. I mentioned the GOAT. We send the GOAT target list, but we also pull some information from GOAT, including estimated genome spans, ploidy, all of that good stuff that we can kind of copy and have at hand for the assembly team so they don't have to keep looking at GOAT. It's all there in the tracking tool. And COPO. So COPO is kind of the entry point for us. COPO is a broker that handles metadata submission to the ENA and to biosamples. And so we can we can connect with Copo via their API, and the two systems can talk together. And we're also adding in support to actually look in the ENA for the biosamples, et cetera, et cetera. So making everything work together. There's also the interactive interface, so you don't have to rely on REST. You don't have to do these API queries. You can use, if you're a registered user with the proper permissions, you can use the admin interface and directly update some of the tables. So project management can go in, sample coordinators, hotspot coordinators, they can go in and edit the information about each of the samples that they're collecting. The genome team can be managed, assigning who's going to do what sequencing center, what assembly group, who's doing the vouchering, barcoding, and kind of copying from the metadata who did the taxonomic identification, who was the sample coordinator, et cetera. Sequencing units can go in and update things, but we encourage them to use the API to do it automatically. This is kind of the main status table. You'll see that I copied some of the style of the Tree of Life portal, right? I thought it was kind of nice. I may regret the decision later, but it works now, but it's in ERGA colors. So the most progressed color is this nice dark blue, and down onto, if you see anything red, there's definitely a problem. So there's filters over here. If I had time, I'd do a demo. You let me know at the end of the call if you want me to open it up. There's filters here, so you can filter by any of the categories, any of the columns here. You can sort by clicking at the top. We now have the sum of the genome spans as a footer in the table. So if you filter for everything that's being curated or past curation, okay, we get about 30 gigs of genome span. Okay, we're supposed to be up to 90 pretty soon. So we're a third of the way there, I would say. There's a little hang up with a few things. High C is very challenging, but I'm not going to go there. I'm just talking about reporting what we see. Yeah, so I mentioned that. And I think I could probably stop here unless we have more time. There is a production instance. That's the one that everyone uses here. It's for the ERGA stream, not for the barcoding stream. So it's called ERGA stream. There's a development server. And if data here gets screwed up, so be it. That's for development. And the code is here. And I do have one person working with me. She's probably on the call. I'm not sure if she's here, Chiara. And Chiara is going to be also developing, and she's the one that is going after people, pestering them to please submit their data. The goal here is to submit early and often, right? As soon as the data is produced, as soon as the reads are produced, they should be submitted to the ENA. This doesn't always happen, and we need to follow up. And the same goes for the assemblies. But I think we're doing pretty well there. We have the EAR process, this review process. And the idea is for me to incorporate this process a little more into the tracking system so that as EARs become available in the GitHub repository, they'll show up here. Okay. And I'll talk with you, Tom, later about how to get that working well. Okay. You can push it to the tracking console, or I can pull it from GitHub. That's it. I just wanted to acknowledge my team members, Fernando and Jessica and Francisco. All now doing assemblies and all doing assembly work, I mean, annotation work as well, and dealing with data submissions. So we used to specialize, but now we need redundancy. We need more people doing assemblies. We need more people handling the data. And hopefully with a tool, the tracking tool that we have here, we can recruit more people to do this, and it will be easy to eventually go cross-institutional. Okay, so that the data is produced and submitted. It can be downloaded from the SRA, from the ENA, and assembled, let's say, in Galaxy. Right. So the idea is to get to a very distributed system. And I think we're getting there. I think it'll be the outcome of the BGE, not necessarily what we have in the beginning, but I'm very happy with how things have been progressing. And thanks to Camila and to Tom and making this not a one-person show. And thanks to Kiara. I don't know if she's on the call, but she's helping out a lot.
Excellent.
That's all.
Thanks very much to all of you.